Hi students, this is Mr. Eckert and we're going to go through Target H11, which says that you should be able to explain the main events in each of the 8 stages of something called meiosis. And I'm sure you remember a process that you learned before called mitosis, and that's going to help you with this process of meiosis because they have a lot of similarities. So just a little bit of review. Remember that normal humans, I'm sure you remember this by now, have a total of 46 chromosomes that are made up of 23 pairs. You got each of those sets of chromosomes, 23, one from mom and one from dad. And they, each of those 23 pairs, if you look at one pair of chromosomes, they're going to be the same size, shape. They're going to have the same genes on them, and that's probably the most important thing. So, of course, the DNA in those two chromosomes are different because one came from your mom and the other came from dad. But those are known as then homologous chromosomes. Homo, again, means the same, so it's not that they have the same DNA, but they have the same genes on them. You may even remember that when you stain them, they all have the same banding patterns, and you saw that when you looked at karyotypes. Now remember, cells that are non-reproductive cells are called somatic cells. So all of those are going to have all 46 chromosomes. And those that do are known as diploid cells because they have all 46 chromosomes. And that you symbolize diploid by using a 2n, n being the number of haploid or haploid chromosomes. Gametes are reproductive cells or sex cells, eggs and sperm. So they only have one set of chromosomes, and that's known as being haploid. Notice that's symbolized by just N. So N is the haploid number for any organisms. So you might know the answer to this question. Why do sex cells only have 23 chromosomes and not 46? And if you think about it, of course, when an egg and sperm fuse during fertilization, that's going to get you back to the diploid number 46, and then that fertilized egg is going to go through the cell cycle, copy its DNA, so you're going to have 46 chromosomes every time that cell then splits and divides. So of the 23 pairs of chromosomes, 22 are called autosomes, and of course one pair determines the sex, and those are sex chromosomes. So the sex chromosomes in humans, you know, are X and Y, XX being for female and XY being for male. So you can see that it's the male or the sperm that determines the sex of an offspring because eggs carry X chromosome only, but a sperm can carry either an X or a Y. So multicellular organisms produce their sex cells through this process called meiosis. And in humans, the eggs are also known as ova. And then, of course, you have sperm. So when an ovum and a sperm combine, you get a full set of chromosomes, half from mom and half from dad. So during meiosis, this process of producing sex cells, the cells divide, and each resulting cell has half the number of chromosomes, so they're haploid again. So the parent cell, the cell that starts the process of meiosis, is a diploid cell, but the daughter cells that result from meiosis are haploid. And there's two main parts of meiosis. There's a meiosis 1 and a meiosis 2. And each part or each phase has 4 stages, and you'll recognize these from mitosis. The PMAT stages, prophase, metaphase, anaphase, and telophase. So in prophase 1, now this is important, homologous pairs of chromosomes will temporarily attach to each other and form this structure called a tetrad. Otherwise, if you remember the main events in mitosis during prophase, it's pretty much the same thing. The chromatin will coil up, and remember, chromatin is condensed DNA. So the chromatin further coils and condenses to form chromosomes, so they become visible during prophase. Once those chromosomes form, again, homologous pairs of chromosomes will join together to form tetrads. The nuclear membrane is going to disappear because the chromosomes are going to start to move around in the cell, and they can't do that with the nucleus there. So the nucleus disappears, and the centrioles start to make spindle fibers, and I'm sure you remember the spindle fibers are going to actually attach to the chromosomes, and that's how they're able to move. Now, this is really important. This only happens in prophase 1 of meiosis. These tetrads, when the homologous chromosomes pair up, you can see here that often pieces of them will literally cross over, break off, and exchange places. So they call that crossing over. You can see in this diagram the blue chromosome, let's say that came from dad, and the red one from mom. After crossing over has occurred, you can see that dad's chromosome has a little piece of mom's chromosome that's now attached. And likewise, mom's has a little piece of dad's chromosome attached. So now, some of mom's DNA is on dad's chromosome and vice versa. So the chromosomes have been recombined. The genes have been recombined. Homologous chromosomes, the alleles, remember alleles are the different forms of a gene. You can find them at the same location. Scientists use the word locus is singular, and loci or loci is plural. So you find those genes on the same location on homologous chromosomes. So the chromatids, sister chromatids, will exchange homologous sections carrying alleles, and that produces recombinant daughter chromosomes with different combinations of alleles. So this genetic variation, this is one of the major sources of genetic variation, and that's a key factor in evolution, which you'll learn about very shortly. All right, so that was prophase 1. Now metaphase 1, remember the tetrads, the pairs of homologous chromosomes, they're going to line up on the equator of the cell. The spindle fibers are attaching, then you can see here, to the centromeres of each of those pairs of chromosomes. So in anaphase 1, the spindle fibers will pull the tetrads apart. And now notice that it's an entire chromosome that's moving towards the opposite end, or the poles of the cell. It says at the end of this phase the chromosome number is now cut in half, but that is not really true until this cell will split. So telophase 1, the cell starts to pinch in, you can see here. And it says the cell usually goes directly to prophase 2. So it goes through PMAT one set of times, so P1, M1, A1, T1. Then it goes through PMAT again, and so you're going to have P2, M2, A2, T2. All right, so when this cell goes all the way through cytokinesis and splits, now you do, of course, have half the number of chromosomes. You'll notice that we started with 4, and now when this cell forms, you're going to have only 2. So meiosis 2. Meiosis 2 is going to look exactly like mitosis. You have 2 haploid daughter cells that were produced by meiosis 1. They're going to divide again, and at the end of meiosis 2, you're going to have 4 haploid cells altogether. In the process of producing sperm, we call this spermatogenesis, and all 4 of those resulting cells are going to survive to produce viable sperm. In women, or girls I should say, it's called oogenesis. That's the production of eggs or ovum. Now you get 4 cells at the end, but 3 of those cells have very little cytoplasm. And 1 cell has a lot of the cytoplasm and will survive to produce 1 viable ovum or egg. And I'm sure you remember that all of this happens when a little girl is born. She has all of the eggs that she's ever going to ovulate in her life. So here's an overview of this process of meiosis. Remember, these are key points. Tetrads are going to form in prophase 1, and crossing over occurs in prophase 1. It's the tetrads, or homologous chromosomes pairs, that line up in metaphase 1, and that's what is separated in anaphase 1. So the daughter cells that result from meiosis 1 are haploid. And during metaphase 2, you get 4 cells at the end, and that produces eggs and sperm. So some differences between meiosis and mitosis. Mitosis doesn't have tetrads because there's no pairing of homologous chromosomes. So therefore, mitosis doesn't have crossing over. That can only happen when you have paired chromosomes. Chromosomes are separated in mitosis while it's tetrads, paired homologous chromosomes that are separated in meiosis 1. In mitosis, the daughter cells are diploid, whereas in meiosis, they're haploid. Mitosis produces more body cells, more somatic cells. In fact, you might remember that this way. Mitosis happens in mito, whereas meiosis produces gametes, sex cells. Here's a diagram showing side by side the two processes. Notice here you start with 2 chromosomes in the parent cell, and then at the end of mitosis you still have 2 chromosomes. In meiosis you start with 2 chromosomes, and notice at the end of meiosis you have 4 cells, each only having 1 chromosome, so the number's been cut in half. Alright, so why does mitosis happen? It happens for growth and for repair. Meiosis, sole purpose is for reproduction. Now, you should be able to identify different phases of meiosis. So it says although there's differences between each, if you can learn the main events, then you'll be able to identify the phase, P, M, A, or T. So in prophase, remember the nucleus will be disappearing, the chromosomes will become visible, or will be visible. In metaphase, the chromosomes will be lined up in the middle. Anaphase, there's going to be a pulling apart either of chromosomes or of tetrads. And remember, in telophase, just look for a pinching in of the cell. Determining whether it's meiosis 1 or meiosis 2 is easy. In meiosis 2, you're going to see 2 cells, always. In meiosis 1, you're only going to see 1 cell. So, if you see 2 cells, it must be meiosis 2. If there's 1 cell that has pairs of chromosomes, you know it's not mitosis, it's meiosis 1. Okay? And if there's 1 cell and it has separate chromosomes and no pairs, it's going to be mitosis. So, look at number 1, and what do you think that is? That's metaphase 2. You see 2 cells, so it's got to be the second phase of meiosis, and you have chromosomes lined up in the middle, so metaphase 2. Number 2, what do you think that is? That's just anaphase of mitosis. Notice what has been pulled apart is chromosomes. So think about what this looked like before this stage. You would have had chromosomes lined up in the middle, and you only have 1 cell. So that's anaphase of mitosis. How about number 3? That's just plain old prophase. You don't see chromosomes in pairs, and you can see the nucleus is disappearing, so that's prophase. How about number 4? Anaphase 1 of meiosis. Notice these are pairs of chromosomes that are being pulled apart. So again, think of what it looked like before this. You would have had tetrads lined up in the middle. So it must be meiosis. And you only see 1 cell, so it's anaphase 1. Be careful on this one. What about this one? That's telophase 1. You might say, well, I thought there's 2 cells there, but technically they haven't formed 2 cells yet. It's still pinching in, but you can see that you have pairs of chromosomes here. So that's telophase 1 of meiosis. So that's it for that target. If you have any questions, please make sure you ask in class.